Humans think about agriculture in a very classical sense. The farmer on his tractor, or even all the way down to working with horses and holding the plow. And so when we're messing with agriculture, with whatever technology, GMO, gene editing, you're tarnishing that image. But the reality is that the technology has really allowed us to make advances, and that's not romantic. We're asking basic questions about the genes that control flower production, because flowers lead to fruits and, of course, seeds. And gene editing was just another tool for us to add to be able to do the basic biology. When CRISPR-Cas9 came, it was immediately apparent that we could create mutations in any gene that we wanted in the genome of tomato. The process, of course, could be looked at from the outside as a foreign, unfamiliar, dangerous, GMO-related process. So the way GMO works is that you're taking a piece of foreign DNA, a gene or another piece of DNA that carries a collection of genes, and you're inserting that into the chromosomes of the plant to give a particular trait that's desirable. CRISPR technology works differently than GMO. With gene editing, you're using a set of molecular scissors, and these molecular scissors allow you to cut or edit the DNA that already exists in the plant. And creating edits and mutations, desirable ones, is very similar to the kinds of existing genes and existing DNA mutations that nature has already generated, and those mutations that have allowed us to improve those crops through breeding.
Gene editing is an unscientific term that sounds nice, but it doesn't really describe the technology. So products like CRISPR are absolutely biotechnology. We would, of course, not allow anything developed with CRISPR to have the Non-GMO Project butterfly on it. We think that it's really important for there to be transparency about how products are developed. The main concerns about genetic engineering, including gene editing, is that it's a very reductionist approach. Small changes to DNA make big changes in the organism. They found a way to create changes in the organism that they intend to make, but it's really beyond current scientific capacity to monitor all of the unintended consequences that happen. There's really no way to ensure that this is safe and healthy to put in the public food supply. So until they can develop a way to provide confidence that this is safe, it should stay in a laboratory.
I do think that as there's more understanding of what gene editing is doing, it's very clear that it has a huge upside. And that huge upside is very different than what was presented as the upside of GMO starting in the early 90s. And that it's really just a tool or a process to create the kinds of changes that you already found in nature that allowed us to have the crops that we eat now. I would hope that CRISPR-produced products, whether it's out of my lab or other labs, would be on the shelves within less than a year, and I think that will happen. The big question is, global population is rising, less arable land, less resources, less water, environmental change. Is CRISPR-Cas9 going to address these problems? It will not be the panacea. I think that when we look at this technology, we have to look at the hope that it brings for us to be able, for example, to breed crops that now might be more tolerant to extreme weather conditions, or that might produce more yield in a static environment. CRISPR-Cas9 gene editing is a powerful technology that simply opens the door to be able to address some of these problems, whereas previously we weren't even in a position to be able to start.